Ever heard of a bacteria that can, like, chill in amber for millions of years
Oh, yeah.
and then wake up like nothing happened?
Yeah.
That's the kind of resilience we're talking about today as we deep dive into the world of Bacillus and his relatives,
It's a fascinating group for sure.
all based on the insights from Principles and Practice of Clinical Bacteriology. Second edition
Great book.
by Gillespie and Hockey.
Yeah, it's a classic. For you, our medical microbiologist listeners, this deep dive,
Absolutely.
this isn't just like a fun fact. It's crucial for understanding how these bacteria can impact your work every day.
For sure.
We'll be focusing on their identification, the infections they cause,
Right.
and how to effectively manage them.
All right, so let's kick things off with how we even classify these microscopic ninjas.
Okay.
I mean, DNA analysis has really shaken up the bacterial family tree, hasn't it?
It really has. The genus Bacillus used to be this massive group with over 140 species all lumped together.
Wow.
But thanks to DNA analysis, we've realized that some of those bacteria were distant cousins at best.
Now, the genus is down to about 80 species,
Okay.
with many former members branching off into new genera like Painobacillus and
Yeah.
Ravibacillus.
So it's like a bacterial soap opera with family secrets and surprise splits.
Okay.
But I imagine this new classification system creates some challenges for microbiologists in the lab, right?
Absolutely. DNA analysis is great.
Yeah.
It gives us a clearer picture of evolutionary relationships, but it doesn't always translate to easily observable differences under a microscope. So you might have two species that are genetically distinct,
Right.
but look almost identical on a petri dish.
Oh, so how do you tell them apart then?
Well, that's the challenge, isn't it? It takes a combination of techniques,
Right.
both traditional and molecular.
So what are the key features that make a bacillus a bacillus? What should our listeners be looking for when they're in the lab?
Good question. Think of your classic bacillus as a rod-shaped cell,
Okay.
often gram-positive.
And most importantly, capable of forming those incredibly durable endospores.
Right.
It's like they have a built-in escape pod for tough times.
And those endospores are seriously tough customers, aren't they? They can withstand heat,
Oh, yeah.
radiation, disinfectants, you name it. It's no wonder these bacteria are found everywhere.
They are the ultimate survivors, that's for sure.
Truly.
But here's a twist that might surprise you. Not all bacillus species are aerobic, meaning they require oxygen to grow.
Oh, really?
We even have some anaerobic members of the family,
Whoa.
which challenges the classic textbook definition.
So you're telling me there are bacillus that don't need oxygen?
That's right. They're out there.
That's like finding a fish that lives on land. It seems like they're always breaking the rules.
They do like to keep us on our toes.
They do.
And it highlights the diversity within this genus. They thrive in a wide range of environments, from hot springs to polar ice caps.
It's amazing how adaptable they are.
We have mesophiles, thermophiles, and psychrophiles, all within the same family.
It's mind-blowing. So, for our medical microbiologist listeners, which species should be on their radar, especially those causing human infections?
Well, the ones you'll encounter most frequently are those that grow happily
Right.
on standard lab media at 37 degrees Celsius.
Okay.
This includes the Bacillus cereus group, which is particularly important for human health.
Okay, so let's talk about the Bacillus cereus group,
Okay.
especially Bacillus anthracis and Bacillus cereus.
Right.
These two have quite a reputation. What makes them stand out from the crowd?
Well, the Bacillus cereus group includes some heavy hitters like B. anthracis,
Of course.
B. cereus, B. mycoides, B. thuringiensis, B. weihenstephanensis, and B. pseudomycoides.
Wow, that's a mouthful.
But B. anthracis and B. cereus are the most significant for human infections.
Right.
And interestingly, they can create colonies with appearances ranging from a ground glass texture to smooth and moist.
Okay.
And some even sport those characteristic Medusa head edges.
Medusa head edges. Now that's a visual I won't forget.
It's pretty distinctive.
It-
But you're right, it can be tricky to tell these species apart, especially B. anthracis from its close relatives.
Yeah, how do you do it?
Well, B. anthracis gives us a few clues.
Okay.
Its colonies tend to be smaller, non-hemolytic, and have a unique, tenacious growth pattern.
Tenacious, like they're digging their heels in and refusing to budge.
Exactly. And under certain conditions, B. anthracis produces a capsule,
Interesting.
a protective shield made of poly-D-glutamic acid
Wow.
that helps it evade the immune system. It's like a microscopic invisibility cloak.
Now that's sneaky. Speaking of sneaky, let's dive into the world of B. anthracis itself.
Okay.
It's a bacterium with a serious reputation And I'm curious to know what makes it so dangerous.
Well, it all starts with the spore.
Okay.
The spore is what makes B. anthracis so dangerous.
Right.
These hardy structures can persist in the soil for decades,
Oh, wow.
often lurking near animal carcasses.
Huh.
When an unsuspecting animal or human comes into contact,
Right.
the spores seize their opportunity.
So it's like a waiting game for these spores, lying dormant until a perfect moment to strike.
Precisely. Once inside the host, they're engulfed by macrophages, those immune cells that usually gobble up invaders.
Okay.
Some macrophages are destroyed,
Right.
but others become unwitting accomplices, transporting the spores deeper into the host's body toward the lymph nodes.
Wait, so the bacteria are hitching a ride inside immune cells? That's like sneaking into a fortress disguised as a friendly soldier.
Exactly. And here's the kicker. Those spores can remain dormant inside the macrophages for up to 60 days.
60 days, that's a long time.
It's a delayed action time bomb.
That's terrifying. What finally triggers them to wake up and cause havoc?
The environment inside the host provides the cues. Elevated carbon dioxide levels and the warmth of body temperature signal to the spores that it's time to spring into action.
I see.
They germinate, shedding their protective coats and unleashing their arsenal of virulence factors, including the deadly anthrax toxin.
So those virulence factors are like the bacteria's weapons, right? Exactly.
And the genes that code for them are conveniently located on two plasmids.
That's right.
PX01 for the toxin and PX02 for the capsule.
You got it. And that capsule we talked about earlier, made of polyOD-glutamic acid, plays a crucial role in helping the bacteria evade the host's immune system.
Right.
It's like a shield that deflects attacks.
So it's not just about producing toxins, it's about evading the body's defenses as well.
Exactly.
It seems like B. anthracis has a well-thought-out battle plan.
It does.
But let's break down the anthrax toxin itself. What makes it so lethal?
The anthrax toxin is a fascinating but deadly three-component system.
Okay.
Composed of edema factor, EF, protective antigen, PA, and lethal factor, LA.
Okay, I'm following along.
Individually, they're harmless,
Right.
but together they form a potent weapon.
So it's all about teamwork for this toxin, like a microscopic demolition crew.
Precisely. PA acts as the scout.
Okay.
It binds to specific receptors on host cells and forms a gateway for EF and LF to enter. So PA essentially unlocks the door for the other two to wreak havoc.
Exactly. Once inside, EF disrupts cellular processes by increasing levels of CAMP.
Oh.
This disruption leads to a dangerous fluid imbalance known as hypovolemic shock.
Oh, wow, that's scary.
And LF is a precision weapon.
Okay.
It targets and destroys key proteins within host cells, especially macrophages. By taking out these vital immune cells, LF cripples the host's defenses.
It's like they're disabling the body's security system,
That's a good analogy.
leaving it vulnerable to attack.
Exactly. The result is a rapid buildup of bacteria in the bloodstream, overwhelming the host's ability to fight back.
Wow, that's both fascinating and terrifying. Now let's shift gears for a bit and focus on B. cereus, the food poisoning culprit.
Oh.
I'm sure many of our listeners have encountered this one in their work.
Probably.
What makes it so good at causing those unpleasant bouts of food poisoning?
B. cereus is notorious for causing two distinct types of food poisoning.
Yeah.
The diarrheal type
Yeah.
and the emetic type.
Right.
The diarrheal type is all about toxins produced by the bacteria in the small intestine after you eat contaminated food that wasn't stored properly.
So it's like a delayed reaction, making you regret that second helping of rice.
You could say that. Symptoms usually hit about 8 to 16 hours after eating the contaminated food.
Okay.
And thankfully, they tend to resolve within 24 hours.
Okay, that's reassuring. But what about the emetic type? How does that one differ?
The emetic type is a true food poisoning caused by a preformed toxin called cerulide
Okay.
that's already present in the food when you eat it.
I see.
This one is all about speed. Symptoms strike quickly, usually within 1 to 6 hours.
So one type makes the toxins after you eat, and the other has the toxins ready and waiting.
Precisely.
Talk about a double whammy. But here's a question that's been bugging me. Cooking kills bacteria, right?
Generally, yes.
So how does B. cereus manage to survive and cause food poisoning even after the food has been cooked?
That's a great question. The answer lies in those incredible endospores we talked about earlier.
Right.
These spores are heat resistant, meaning they can survive even high cooking temperatures.
Oh, wow.
So even if you thoroughly cook your food,
Yeah.
those sneaky spores can linger,
Right.
waiting for the opportunity to germinate and cause trouble.
Gotcha.
Especially if the food isn't stored properly.
It's like they're playing a microscopic game of hide and seek.
They are.
So it's not enough to just cook the food.
Right.
We need to be vigilant about proper storage as well.
Absolutely.
So what else should our listeners know about the toxins produced by B. cereus?
Well, Bacillus doesn't just rely on one or two toxins to cause its mischief. It has a whole arsenal. We've got HBl, Ne, BSET, ENTFM, ENTK, and of course cereulide.
Wow.
Each playing a role in causing those unpleasant gastrointestinal symptoms.
Wow, that's quite a lineup. 6 different toxins. It's like B. cereus came prepared for a toxin showdown?
They did.
I'm particularly intrigued by cereulide, the emetic toxin. What makes it so unique?
Cereulide is a fascinating toxin with a complex chemical structure known as a dodecadepsipeptide. A dodecadepsipeptide. It's a mouthful, I know.
It is.
But the important thing is it's incredibly resistant to heat and breakdown by enzymes, making it a formidable opponent.
So even if you reheat your food, cereulide can still pack a punch.
You bet. And while the exact mechanism of how it causes vomiting is still being studied, we know that it involves the vagus nerve, which connects the brain to the gut.
So it's like cereulide is sending a direct message to the brain saying, get this out of here ASAP.
That's a good way to put it.
It's a powerful toxin that can leave you feeling quite miserable.
No doubt about it.
Well, this has been a fascinating overview of the Bacillus genus and its mischievous members.
Yeah.
We've covered their unique characteristics, their sneaky survival strategies, how they interact with their environment and their hosts. But I'm curious, where do these bacteria hang out in the environment and how do they spread to humans?
Yeah.
It's all about ecology and epidemiology, right?
You've got it. And it really depends on the species.
Okay.
Let's start with B. anthracis.
All right.
This one's an obligate pathogen, meaning it needs a host to survive.
Okay.
It's primarily a disease of herbivores like cattle and sheep, and its spores can persist in the soil for decades.
Wow.
Especially in areas where infected animals have died.
So it's like a grim cycle of infection lurking in the soil waiting for a new host to come along.
Unfortunately, yes.
That's a bit unsettling.
It is.
And humans typically get infected through contact with infected animals.
Right.
Or contaminated animal products.
Exactly.
It's a reminder that we're all part of a complex ecosystem.
That's for sure.
And these bacteria can easily jump from animals to humans.
They can.
A sobering thought.
It is.
Now let's contrast that with other Bacillus species.
Okay.
They seem to be everywhere, like they've mastered the art of survival.
They have. Unlike B. anthracis, these Bacillus species are ubiquitous. They're found in soil, dust, water, air, you name it. And their secret weapon, once again, is this incredible endospores.
Those endospores are like microscopic survival pods.
They are.
Allowing them to withstand even the harshest conditions.
Right.
It's no wonder they're so successful at colonizing diverse environments.
Exactly. And while most Bacillus species aren't inherently bad guys, they can cause opportunistic infections, particularly in people with weakened immune systems.
So they're more likely to cause problems in individuals who are already vulnerable.
Precisely. It's a reminder that our immune system plays a crucial role in keeping these ubiquitous microbes in check.
This has been an incredible journey into the world of Bacillus so far.
It has.
We've covered their unique characteristics, their sneaky survival strategies, and how they interact with their environment and their hosts.
We've covered a lot of ground.
We have. I'm ready to put on my medical microbiologist hat and delve into the clinical aspects of these infections.
Excellent. That's where we'll pick up in the next part of our deep dive. We'll explore the clinical features of Bacillus infections, how to diagnose them in the lab, and what strategies we can use to effectively manage and prevent these infections. Stay tuned.
Sounds good.
Yeah.
Welcome back to our deep dive into the world of Bacillus. Last time we explored their unique characteristics, their sneaky survival strategies, all that good stuff.
Right.
Now let's switch gears
and
focus on what you, our medical microbiologist listeners, need to know to tackle these infections head on.
Sounds good.
Perfect. I'm ready to put on my lab coat and gloves. Let's start with Bacillus anthracis.
Okay.
The one with a, scary reputation.
Yeah.
What kind of clinical presentations should we be prepared for?
Bacillus anthracis is like a master of disguise.
Okay.
It can manifest in several ways,
Right.
depending on how it enters the body.
Okay.
The most common form is cutaneous anthrax, which happens when spores get into the skin through a cut or abrasion.
So this is the classic presentation that gives anthrax its name.
Yes.
Anthrax actually comes from the Greek word for coal.
Right.
Because of that characteristic black eschar that forms on the skin.
It is.
It's a pretty distinctive sign, isn't it?
It is, yeah.
Yeah.
It usually starts as a small, itchy bump that quickly develops into a ring of blisters. Then the lesion ulcerates, dries up, and blackens, forming that telltale eschar.
It sounds pretty straightforward, but are there any other conditions that could mimic those symptoms?
Absolutely.
Leading to a misdiagnosis?
There are other skin infections that might resemble cutaneous anthrax initially,
Right.
but the appearance of the eschar, combined with a history of potential exposure to infected animals or animal products, is a strong indicator.
So it's all about piecing together the clues. And how serious is cutaneous anthrax? Is it always life-threatening?
Well, the good news is that most cutaneous anthrax infections are self-limiting and respond well to treatment.
That's good.
However, the swelling around the lesion, known as edema, can be extensive. In rare cases, particularly with lesions on the face or neck, the edema can even obstruct the airways.
That makes sense.
So early diagnosis and treatment are key, even if most cases are relatively mild.
Absolutely. So early diagnosis and treatment are key, even if most cases are relatively mild.
Right.
Now let's talk about inhalational anthrax.
Okay.
This form is much less common than cutaneous anthrax, but far more dangerous.
Much more dangerous.
It occurs when someone inhales spores,
Right.
which then travel to the lymph nodes in the chest.
Right.
This is the type we often hear about in the news, the one associated with bioterrorism.
Right. Unfortunately, yes, and that's what makes it so concerning.
Yeah, I can see why.
Inhalational anthrax starts with flu-like symptoms, making it really tricky to diagnose early.
So it's a bit of a wolf in sheep's clothing, starting with mild symptoms that could easily be mistaken for something else.
Exactly. But as the infection progresses, it can lead to severe respiratory distress, shock, and even death.
That's scary. It really highlights the importance of a thorough patient history.
Oh, yeah.
Especially when we're dealing with potential bioterrorism threats.
Absolutely. We need to be vigilant and consider all possibilities.
Yeah.
Now, while less common than the cutaneous and inhalational forms, we also have gastrointestinal anthrax.
Okay.
This can occur when someone eats meat from an animal that died of anthrax.
So this is more likely to occur in regions where people might consume meat from animals that haven't been properly inspected or that have died from unknown causes.
Exactly. The symptoms can range from mild gastroenteritis to severe systemic illness.
Wow.
And unfortunately, the fatality rate can be quite high.
It seems like anthrax is a real chameleon presenting in so many different ways.
It is.
Is there any other clinical presentation we should be aware of?
Well, meningitis is a serious complication that can develop from any form of anthrax. It's a rapidly progressing infection that affects the lining of the brain and spinal cord.
All right.
And the prognosis is often poor.
Okay, that's a lot to keep in mind for anthrax.
It is.
It's certainly a bacterium that demands our respect.
It does.
Now, let's switch gears again and focus on Bacillus cereus, the food poisoning culprit. What kind of clinical features are we looking for with this one?
With Bacillus cereus, the clinical picture is usually less dramatic than anthrax.
Yeah, I imagine so.
But it can still be quite unpleasant for those affected. As we discussed earlier, there are two main types of illness, diarrheal and emetic.
Let's start with the diarrheal type.
Okay.
What are the telltale signs that someone might be dealing with Bacillus cereus diarrhea?
The symptoms typically include abdominal cramps and watery diarrhea, often accompanied by a feeling of needing to strain, which is known as rectal tenesmus.
So basically a really bad case of the runs.
You could say that. The symptoms usually develop within 8 to 16 hours after eating contaminated food. And thankfully, tend to resolve within 24 hours.
Okay, that's good to know. Most cases seem to be self-limiting.
They are.
But what about the emetic type?
Yeah.
How does that one present?
The emetic type is characterized by nausea and vomiting, much like staphylococcal food poisoning. The symptoms usually straight quickly within 1 to 6 hours after eating the contaminated food.
So with the emetic type, you know pretty quickly if you've ingested that preformed toxin, cerulide.
Exactly.
What about the duration of the symptoms? How long does this unpleasant episode typically last?
Fortunately, the symptoms usually subside within 24 hours.
Okay, good.
However, it's worth noting that in rare cases, cerulide has been associated with severe liver complications.
That's concerning. So even though Bacillus cereus food poisoning is generally considered mild and self-limiting, it's important to remember that it can have more serious consequences in some cases.
They can.
You're absolutely right. We can't let our guard down. And it's also important to remember that Bacillus cereus can cause other infections besides food poisoning.
Oh, yeah.
Especially in people with underlying medical conditions or weakened immune systems.
Absolutely.
Okay, let's talk about those other infections.
Right.
What else can Bacillus cereus do besides wreak havoc on our digestive systems?
Well, one particularly concerning infection is Bacillus cereus endophthalmitis, a severe eye infection that can occur after trauma or surgery.
So this is something our ophthalmologist colleagues need to be aware of.
They do.
Potential complication after eye surgery or an injury.
Exactly. Bacillus cereus endophthalmitis is a rapidly progressing infection that can lead to vision loss.
Oh, wow.
And even the loss of the eye if it's not treated promptly.
That sounds incredibly serious.
It is.
What other infections have been linked to Bacillus cereus?
It seems like it can cause problems in various parts of the body. We've seen it cause a wide range of infections, including bacteremia, meningitis, wound infections, pneumonia, and even endocarditis.
It's amazing how a bacterium that causes a relatively common food poisoning can also lead to such serious infections. It really highlights the importance of not underestimating these microbes.
I completely agree. And speaking of not underestimating them, we should also mention that other Bacillus species, besides Anthracis and cereus, can be opportunistic pathogens as well.
So it's not just those two big names we need to be on the lookout for.
That's right. Species like Bacillus licheniformis, Bacillus subtilis, and even some Haenobacillus species have been implicated in everything from eye infections to bacteremia and meningitis. It's a diverse group with a wide range of pathogenic potential.
It seems like the Bacillus genus is full of surprises. They're everywhere.
They are.
They're resilient.
Right.
And they can cause a wide range of infections.
Right.
Now, I'm curious, how do we actually identify these bacteria in the lab? What are the key steps in laboratory diagnosis?
Well, the first step is to recognize that we're dealing with a Bacillus species. This involves observing their characteristic rod-shaped morphology under a microscope, confirming the presence of those telltale endospores, and performing a gram stain.
So we're back to the basics of microscopy and staining techniques.
Exactly. Those classic techniques are still incredibly valuable.
Absolutely.
And don't forget about the endospores. They're crucial for identification because they're larger, brighter, and more uniform in shape and position than other cellular inclusions. They really stand out under the microscope.
It's like spotting a tiny bacterial pearl.
It is.
Now, once we've confirmed that it's a Bacillus species, how do we narrow it down to the specific culprit?
Good question.
There are so many different species, and they can look quite similar.
They can. There are several approaches we can take. Traditional phenotypic tests, like those outlined in Berger's manual, can be helpful for identifying specific metabolic characteristics. But they can be quite time-consuming.
So if we need a faster turnaround time, what are our options?
Commercial identification systems like API and Vitek are a great option. These systems use a combination of biochemical reactions to create a unique profile for each species, like a bacterial fingerprint.
That's fascinating. So we're essentially analyzing their metabolic signatures to figure out who they are.
Exactly.
What about molecular methods?
Yes.
Are those playing a role in Bacillus identification as well?
Absolutely. Molecular methods like PCR are becoming increasingly important. They allow us to quickly detect specific genes, especially those associated with virulence factors.
That makes sense, especially when we're dealing with a potential bioterrorism threat like anthrax.
It does.
We need to know as quickly as possible what we're dealing with.
Exactly. Speed and accuracy are crucial in those situations.
Absolutely.
Now, for Bacillus anthracis specifically,
Okay.
there are some additional tests that are essential for confirmation.
Okay, let's talk about those tests.
Right.
What makes Bacillus anthracis stand out in the lab?
Yeah.
What are the unique features that allow us to say, aha, this is definitely Bacillus anthracis?
Well, one key characteristic is its susceptibility to the gammaphage, a virus that specifically infects Bacillus anthracis. This is a very specific test that can help us quickly rule in or rule out the anthracis.
So it's like Bacillus anthracis has a kryptonite, a weakness that we can exploit to identify it.
Exactly. Another unique feature is its sensitivity to penicillin, which is unusual for a Bacillus species.
Right.
Most other members of the genus are resistant to penicillin, making this a helpful diagnostic clue.
So it's like Bacillus anthracis is playing by a different set of rules.
It is.
It's susceptible to a drug that most of its relatives can shrug off.
Exactly. And of course, we can't forget about the capsule.
Right.
Virulent strains of Bacillus anthracis produce a distinctive capsule that can be visualized using special staining techniques, like amphibian stain.
So we're looking for a combination of phage susceptibility, antibiotic sensitivity, and that unique capsule to confirm that we're dealing with Bacillus anthracis.
Exactly.
What about Bacillus cereus?
Okay.
Are there specific tests we use to confirm its presence, especially in cases of food poisoning?
For Bacillus cereus, we have specific assays that can detect the various toxins it produces, particularly the diarrheal and emetic toxins.
So we can actually test for those toxins directly?
We can.
Either in food samples or in clinical samples from patients?
Yes, we can. These toxin assays are especially helpful in confirming cases of Bacillus cereus food poisoning and in investigating outbreaks.
This has been a fascinating tour of the laboratory techniques used to diagnose Bacillus infections.
Yeah.
It's amazing how we can use these tools to pinpoint the culprits and understand what we're dealing with.
It is.
But now that we've identified the enemy, let's talk about how to fight back.
Excellent. That brings us to the crucial topic of treatment and management.
Right.
We'll discuss the antibiotics that are effective against different Bacillus species, the challenges of antibiotic resistance, and the strategies we can use to prevent these infections from occurring in the first place.
Okay.
Stay tuned.
Welcome back to the final part of our deep dive into the world of Bacillus. We've explored their unique characteristics, delved into their clinical presentations, and learned how to identify them in the lab.
Right.
Now let's talk about the battle plan. How do we effectively manage and prevent infections caused by these resilient bacteria?
That's right. Let's equip you with the knowledge and strategies to combat these microscopic foes.
And when it comes to managing infections, antibiotics are usually our first line of defense, right? But earlier we touched upon the looming threat of antibiotic resistance.
Yeah.
How did that impact our approach to treating Bacillus infections?
Antibiotic resistance is a major challenge.
Yeah.
And it's something we need to be aware of when choosing the right treatment for Bacillus infections.
Right.
Let's start with Bacillus anthracis.
Okay.
Historically, penicillin has been the go-to antibiotic, and it's still effective against most strains.
So penicillin is still a good choice, but we need to be mindful of resistance.
Exactly. We're seeing more and more reports of penicillin-resistant Bacillus anthracis strains.
Oh.
So it's crucial to perform susceptibility testing to ensure that the strain we're dealing with is actually susceptible to penicillin.
That makes sense. We can't just assume that penicillin will work every time.
No, we can't.
So what are our alternatives if penicillin isn't an option? What other antibiotics can we use to combat anthrax?
Well, there are other effective antibiotics, including tetracyclines like doxycycline and fluoroquinolones like ciprofloxacin. These antibiotics target different pathways in the bacteria, making them good alternatives when penicillin resistance is suspected.
So we have backup options. But I've also heard about combination therapy being used for anthrax.
Yeah.
What's the rationale behind that approach?
Combination therapy, using 2 or more antibiotics simultaneously, is often recommended for severe anthrax cases.
Okay.
Or when we're concerned about potential bioterrorism. The idea is to hit the bacteria with multiple drugs, making it much harder for them to develop resistance.
It's like overwhelming them with a multi-pronged attack.
Right.
Making it much more difficult for them to survive.
Exactly.
And speaking of bioterrorism, we also talked about post-exposure prophylaxis.
Yeah.
Can you remind us what that is and why it's important?
Post-exposure prophylaxis or preventative treatment is crucial for individuals who have been potentially exposed to anthrax spores, especially in a bioterrorism scenario.
Okay.
Ciprofloxacin or doxycycline are the recommended antibiotics for this purpose.
So if someone's been exposed to anthrax, taking these antibiotics can prevent the infection from taking hold. It's like giving their immune system a head start in the fight.
Exactly. It's a preventative measure to reduce the risk of developing anthrax.
Okay.
However, it's important to emphasize that early diagnosis and treatment are always the best course of action.
Right.
If someone develops symptoms of anthrax, we need to start treatment as soon as possible.
That makes sense. Time is of the essence when dealing with a potentially serious infection like anthrax.
Absolutely.
Now, what about Bacillus cereus? How do we typically manage infections caused by this bacterium?
Bacillus cereus is a bit trickier because it's naturally resistant to penicillin and many cephalosporins.
Oh, really?
This resistance is due to its ability to produce beta-lactamases, enzymes that break down these antibiotics.
Those beta-lactamases are like tiny antibiotic shredders, aren't they?
That's a good way to put it.
They make those drugs ineffective.
They do.
So what are our options for treating Bacillus cereus infections if those common antibiotics are off the table?
Well, we often turn to antibiotics like clindamycin, erythromycin, vancomycin, and aminoglycosides.
Okay.
These antibiotics have different mechanisms of action and are not affected by beta-lactamases, making them effective against Bacillus cereus.
So we have alternatives, but it seems like susceptibility testing is crucial in this case.
It is.
To ensure that we're choosing the right antibiotic for a particular strain of Bacillus cereus.
Absolutely. We need to make sure that the antibiotic we choose is actually going to kill the bacteria we're targeting.
It's all about personalized medicine for bacteria.
It is.
Tailoring the treatment to the specific bug we're dealing with.
Exactly. And for other Bacillus species, the treatment approach is similar to what we do for Bacillus cereus.
Okay.
We need to be mindful of potential resistance mechanisms and use susceptibility testing to guide our therapy choices.
So it's a good rule of thumb to always perform susceptibility testing when we're dealing with Bacillus infections, even if we think we know what we're dealing with.
Absolutely.
Now, let's switch gears and talk about prevention.
Okay.
How can we reduce the risk of these Bacillus infections in the first place? What can we do to stop them before they start?
Prevention strategies really depend on the species we're talking about. For Bacillus anthracis, the focus is on controlling the disease in animals, primarily through vaccination.
So by protecting animals, we're also protecting humans. It's all connected.
Exactly. There are also vaccines available for humans who are at high risk of exposure, such as military personnel, veterinarians, and certain laboratory workers.
That makes sense. We need to protect those who are on the front lines. But what about preventing those pesky Bacillus serious food poisoning incidents? I know I'm guilty of leaving leftovers out on the counter for a bit too long sometimes.
We've all been there.
Yeah.
The key is proper food handling and storage. We need to cook food thoroughly, especially meat and rice, to kill those heat-resistant spores.
Okay.
And we need to refrigerate leftovers promptly to prevent those spores from germinating and multiplying.
So no more late-night snacking on cold rice.
Not unless it's been properly refrigerated. Those spores are just waiting for the opportunity to strike.
They are sneaky.
And in healthcare settings, good hygiene practices, such as frequent hand washing and proper sterilization of equipment, are essential for preventing opportunistic Bacillus infections, especially in patients who are immunocompromised.
Those are all great tips for staying safe from these ubiquitous bacteria.
Yeah.
I'm feeling much more knowledgeable about the Bacillus genus now.
I'm glad to hear that. It's always exciting to delve into the world of microbes and uncover their secrets. They play such an important role in our lives, both for good and for bad.
And that brings us to the end of our deep dive into the world of Bacillus. We've explored their unique characteristics, their ability to cause a range of infections, and the strategies we can use to diagnose, treat, and prevent these infections. But remember, the world of microbiology is constantly evolving.
It is.
New discoveries are being made all the time, and our understanding of these microbes is constantly changing.
That's for sure.
That's why it's crucial to stay curious, keep learning, and be adaptable. The more we learn about these microbes, the better equipped we'll be to protect ourselves and our patients from their harmful effects.
Couldn't agree more.
So to all our listeners, keep exploring, keep learning, and never stop asking questions. The world of microbes is full of wonder and challenges, and we're all in this together. Until next time, Stay safe and stay curious.